So welcome everyone to the next session. We have Jonathan just ready to go, and Carmen is going to introduce, so I will disappear. Take it away, Carmen.
Okay, well, Jonathan, thank you for accepting my invitation. Dr. Rubin is professor and chair of the mathematics department at the University of Pittsburgh. He is focused on understanding how the intrinsic dynamics of network elements interact with the architecture and property of coupling to drive network activity. I also happen to know he's an expert on fast-slow analysis. I don't know if you'll be talking about any of that today, but take it away, Dr. Rubin.
Yeah, thanks a lot, Carmen, and all of the organizers. It's great to present here. I'm looking at my slides, as I guess those of you who have presented know, so I don't see anything else going on, but I'll focus on the slides and then be happy to answer questions afterwards. So I'm going to talk about respiration. And when we learn about the respiratory system, in biology, it's sort of presented as this collection of tubes and bags, right? Tubes and sacs, this very mechanical structure, together with a chemical component for exchange of, gases between the lungs and the air that we're breathing in. But really, all of this mechanical structure just sits there inert without some sort of neural signals to drive the muscles to put this thing in motion. And so really the respiratory system, from a global perspective, is this combined structure that has mechanical components as well as all of these neural components that have been worked out in some detail in mammalian brainstem. And the neural components, give these outputs that eventually go through motor neurons and drive the muscles to actually allow us to breathe, which is different from cortical coding or something like that, but certainly fundamental for survival. So in this talk, I'm going to be focusing on two aspects of the neural component of the respiratory system. One, which will be in the second part of the talk, will be these interacting populations of neurons that are responsible for the full respiratory rhythm. And in the first part of the talk, I'm going to zoom in on this excitatory kernel of what are called pre-inspiratory and inspiratory neurons in the pre-Butzinger complex that Carmen mentioned in her talk. And these guys are fairly unusual in this system because they're excitatory, and in fact, their output is the output that directly drives the diaphragm to allow us to breathe in, to inspire, hence the pre-II name. Okay, so these neurons are quite interesting and have been the attention of a lot of mathematical studies. The sort of classic model in this area was developed by Butera, Smith, and Rinzel in the late 90s. And so this is, as Carmen also mentioned, a square wave bursting model. And so you can see these are actually experimental traces, but the model does this as well. These neurons, if you inject current into them, they go from quiescent to bursting, and the burst properties depend on the current level until eventually they can switch over to a tonic spiking state. So we think of these as conditional bursters. They are excitatory. They signal each other with excitatory synapses. And interestingly, in the baseline state, if you decouple them, there's a combination of these different behaviors, Q, B, and T, among the neurons sort of intrinsically within the population. And so again, the overview of my talk is I'm going to talk first about some recent ideas about how the activity organizes across the pre-Butzinger complex, despite this heterogeneity in underlying intrinsic dynamics. And then I'm going to turn to the larger rhythm generating network and talk about, what happens when you take this rhythmic, this kernel that's already rhythmic in its own right and embed it in the more complete CPG network. All right, so part one. The classic idea of what happens in this heterogeneous network is shown in this plot also from the original Butera paper. And so you can see, based on the heterogeneity observed experimentally, they set this simulation up with heterogeneity in model parameters, and these are Hodgkin-Huxley type neurons. And so intrinsically, about 30% of them were bursting. Some you can see are quiescent, some are tonic, right? This is neuron number along the vertical and time along the horizontal, so it's usual spike raster. And then as a whole, if they do some sort of counting of spikes per time bin, the network activity is sort of disorganized and would be insufficient to drive the diaphragm. If you couple these guys together with synaptic excitation, either all-to-all or randomly with a high enough connectivity, then most of the network can get recruited into bursting, and so that produces this sort of network output, which can drive the diaphragm. Okay, so that seems like a pretty simple story. The coupling is enough to overcome the heterogeneity, and even though the onset of activity isn't fully synchronized in individual neurons, that's enough to get population bursts. Okay, but some experimental results call that idea into question. So here we see results from the Feldman lab showing both a measure of the overall activity in the pre-Botzinger complex, so the neurons I'm talking about, in black, along with this output in gray, right? So this is a hypoglossal nerve that's being driven by pre-Bot output. And you can see that depending on the external potassium concentration in the experiment, which they vary by bathing their brain slices in solutions of different potassium concentrations, you get more and more or fewer and fewer of these small events, which occur in the pre-Bot but do not drive the hypoglossal output. Okay, so these have been dubbed as burstlets, as opposed to the bursts that actually do produce population output. And so you can see overall in the pre-Bot, the frequency of these events, whether burstlets or bursts, stays relatively constant over the different experimental conditions. But the hypoglossal output becomes, has very different periodicity depending on the potassium level. And likewise, the amplitudes of the events split into these sort of two clusters, the burstlet events and the burst events. Here's just a summary showing how increasing the external potassium causes the burst low amplitude fraction, so the burstlet fraction, to go away. Normally, the experiments are done with this elevated external potassium concentration, but in fact, in more in vivo-like conditions, there's this mix of bursts and burstlets. Okay, so if we look further into the data about these two these two sort of structures, we see that there's a collection of neurons that are active during both burstlets and bursts, but they show enhanced spiking during bursts. So here are some histograms of the spike counts. There are also some neurons that are only active during the bursts. Okay, so the burstlet neurons are on the left, but there's this additional population that are not active during burstlets that do get recruited during bursts. However, when you compare bursts and burstlets, they do agree, if you look at a spike rate versus time, they do agree almost exactly up until some moment when it's like a decision is made and either the additional neurons are recruited into the burst and the burstlet neurons pick up their activity correspondingly, or the burst fails and we just get a burstlet. Feldman and Cam introduced this, what they call the burstlet hypothesis, as a alternative to the burst hypothesis. So in the Butera work, right, the burst is an essential component of the rhythm. If you somehow block whatever's causing the bursting, you don't have a rhythm at all. The whole rhythmicity falls apart. In the burstlet hypothesis, it is the burstlets that are the rhythmic events, and then there's some other mechanism that sort of decides whether burstlets are recruited to become full bursts or not. Okay, so that's a compelling idea based on the data, but there isn't until now, really a mechanistic theory for how this burstlet and burst idea would work. Okay, so in fact, we had, what I would say is a relatively simple idea that we think it very effectively captures this recruitment. Okay, and the key observation is that the actual motor output, so whether the hypoglossal nerve is recruited or not, depends on a calcium-activated non-selective cation channel. Okay, so an ICAN. So if we interfere with ICAN, for example, by applying cadmium onto a brain slice, you can see that the burstlets disappear. The bursts disappear in the prebot, and we only get burstlets. So disrupting ICAN compromises the recruitment into the full bursts. This is a simulation from a model that my postdoc Ryan Phillips produced to study ICAN during his PhD work, and you can see the same effect in the model. Okay, so what we did in our current work is we built a fairly detailed model of pre-Botzinger complex neurons where we include a calcium-induced calcium release from internal stores. Okay, so if we give our single neuron model a periodic calcium current input, okay, then what this does is it may or may not cause a nonlinear response in terms of a calcium surge on each input cycle, right? So what I'm showing here is the input in green and the actual calcium level inside the cell, which is influenced by this release from stores in blue. And you can see that depending on the amplitude and the frequency of the input, we may or may not get actual large calcium concentrations, and we may get these with sort of different regularities over the course of the input. So very small changes in the input characteristics can lead to this nonlinear amplification of the calcium level in the postsynaptic cell. Okay, so we think this can be the basis for the recruitment mechanism. So we want to implement this in a more detailed way in an actual network of neurons. And to do that, we have to ask, okay, what is really the source of this calcium input, right? Here we just sort of made it up. We just put it into the neuron. And so this is what Ryan had studied with his collaborators in the past. There are two possible ways that you can actually get this calcium coming into the postsynaptic cell. Okay, one is through voltage-gated calcium channels, and another is through the calcium that's associated with synaptic inputs. And if you think about it, the first of these only causes a change in frequencies of the bursting population, because you have to already be active for this voltage-gated calcium to kick in. Okay, when you're active, you get your voltage-gated calcium influx, which activates ICAN, which will speed up the bursting. The synaptic mechanism, on the other hand, allows you to recruit more cells. Okay, the cells that aren't active still will get synaptic calcium influx, which can activate their ICAN, which can recruit those cells. So when you fit a model that has both of these types of calcium sources to experimental data showing how amplitude and frequency of events depend on ICAN, you see that, in fact, the prediction is that 95% of the calcium coming in is synaptic. So we use those numbers, and we built first a 2-neuron network. So we have a pacemaker, which is in blue, and we have this follower, which is in red. And so we change the persistent sodium conductance to distinguish between the two. As a side note, our pacemaker produces this nice ramping dynamics, which is different from standard square wave bursting on its own. And you can ask me about the mechanism for that, which is a paper under review right now in the questions if you want. But you can see that we get a similar effect by varying the excitability of the cells and the weight of the synapses between the two neurons. We can vary how the follower neuron, which is the one with the ICANN effect, is recruited as we move through this parameter space. So we get these different entrainment frequencies as we move through the parameter space. And sure enough, when we build a large network, so we have a population of rhythm-generating neurons, these blue guys down here, and we have this population of follower neurons, and we adjust the synaptic connectivities based on various information in the literature, we can see that we can get these alternating burstlets and bursts depending on whether we get recruitment of the follower through the buildup of the calcium inside those cells, causing this nonlinear calcium-induced calcium release. And so as we vary the excitability of the neurons, which is very much like varying G-tonic in this picture, we can get these different burstlet fractions. Now under the hood, remember in the experiments, it was varying K-bath that caused this change of excitability. So we're doing that. I just don't have time to go into all the details. Okay, but so this would correspond to increasing the potassium concentration in the bath to switch us from a burstlet regime to a full bursting regime. Okay, so I think I have a little bit of time left, I hope. So I'm going to switch over to part 2. Here's Ryan, and here's what I showed you in part 1, that we can put the burstlets and bursts idea is an interesting theoretical concept, but you still have to implement it, right? So you still need to say, what is the rhythm generation mechanism? What's the recruitment mechanism? And we have a persistent sodium dependent burst generation in the burstlet neurons here, very much like the Butera model, and this new idea of the synaptically triggered calcium release leading to the recruitment into bursts or not. Now, there are other factors that I don't have time to go into related to the network spread of the activity and delays in the emergence of the burst related to this idea of percolation. Okay, so now we can ask what happens when the prebot is embedded into the full CPG. So now we have all of these inhibitory populations. This is an all-to-all, three-way network. And so this is why we have inspiration and expiration, right? Not just inspiration, and in fact, it's a 3-phase rhythm, not just a 2-phase rhythm when we breathe normally. Okay, so what we do in this study, we actually simplify things, and so we're just going to represent each one of these populations as a sort of conditional relaxation oscillator. So non-spiking units. We're imagining this is like an average voltage of a synchronized population. Okay, and so when you just focus on the prebot units, as many of you know, right, as you vary the drive, so a synaptic excitation parameter, you're going to change the shape of the voltage nullcline, switch these guys from being quiescent to oscillatory with a middle branch intersection to tonic, the stable fixed point at an elevated voltage. And you can see the bifurcation diagram here as a function of that drive parameter. So we have an oscillatory regime and a tonic regime. Okay, so now we have this population together with these non-oscillating inhibitory populations. And when we couple the network together through the escape and release mechanisms that Carmen mentioned, we get a model rhythm. Okay, so this is what the output of each one of these 4 populations looks like. We have a pre-I, early I component, followed by a post-I, followed by this ramping aug-E, and then we switch over to our next inspiration. And so as we vary the drive to the excitatory kernel, which we saw could switch its intrinsic dynamics completely from quiescent to oscillatory to tonic, that essentially has no effect on the network rhythm. Okay, and we can explain this using moving nullcline analysis. Okay, and I believe my time is probably short, so I'm not going to say too much about this, but let me try to walk you through a little bit of it. Okay, so the idea is when the prebot neurons are tuned to be oscillatory, right, that means on their own they have this nullcline configuration. See the blue here and the red, and so they intersect on the middle branch. So this would be intrinsically oscillatory. There is a knee, right, where the neuron would jump up, and the fixed point is on the middle branch. Okay, now as we give more and more inhibition to these units, okay, the nullcline moves, and eventually the fixed point moves onto the left branch. Right, so we get a fixed point curve, and we also have a corresponding curve of knees. Okay, and so we can plot the level of inhibition versus the H variable, right, the slow variable of these neurons. And we have this curve of fixed points that I mentioned, the curve of knees, and then we're going to get a jump up when we cross the curve of knees. Okay, and forget about the dashed curves, but the solid curve shows that as inhibition wears off, okay, because of the adaptation of the post-I neurons, which are inhibiting these guys, eventually these neurons are able to cross the curve of knees and jump up to the active phase. Okay, when they're tuned to be intrinsically tonic, okay, they no longer have knees eventually, but it doesn't matter because under the influence of inhibition, that intrinsic dynamics is masked and you get the exact same picture. So in terms of dynamics or dynamical mechanisms, these two intrinsic tunings make no difference, and the neuron is able to be released and jump up in the exact same way. Okay, so similarly, if we experiment, a big deal in this field is to gradually block persistent sodium and see what that does to the bursting of the network. And we can look at the period, amplitude, expiratory duration, inspiratory duration. Okay, the actual bifurcation where the prebot neurons change from oscillatory to tonic is along this red curve. And you can see that none of these plots care about the red curve. There's no jump, there's no disruption when you move across the red curve. The tuning just continues smoothly right across. Okay, now here I have two examples where I have varied the drive to each population separately. And I'm looking at these period, inspiratory duration, expiratory duration as a function of drive. And here I have the prebot tuned in these three plots to be oscillatory. And here it's tuned to be tonic. Okay, so I'm going to toggle back and forth. Other than the fact that I use thick lines or thin lines, right, there's basically no effect on the tuning, depending on how you tune the drive to the prebot neurons, so whether they're oscillatory or tonic. Similarly, as you vary the inhibition, either to the prebot neurons or to the populations that are the other, inhibitory populations that are not part of inspiration, you get qualitatively similar effects comparing C to D and A to B on the period and amplitude of the output as you vary the inhibition. Okay, I'm not going into the mechanisms here, but they're all based on this same type of moving nullcline analysis that I went through earlier. Okay, so my final slide then is to conclude that what we see here is that, in fact, the network level outputs persist across the tunings of this embedded kernel. So it's a huge controversy in the field, whether this kernel is really tuned to be intrinsically oscillatory and bursting or not. And this result sort of argues like, hey, that doesn't matter that much. What really matters is that it has the capability of being recruited to burst if it needs to be. Okay, and the reason is that the synaptic signals in the network actually swamp out the intrinsic dynamics when you wire things up. However, there's a hidden important point here, which is the fact that this kernel can function across oscillatory and tonic modes actually does extend the range of network output amplitudes and periods. So basically it makes our breathing more robust and more tunable and more functionally dynamic. So now putting these two works together is for the future. Once we have the prebot actually updated to have the sort of burstlet neurons and the followers, how much of this embedding result will carry over, and that's to be determined. So thanks for your attention. I hope you all have been with me. And now I'm going to reduce this so I can see what's going on.
So thanks a lot, Jonathan. Excellent talk. Just in terms of timing, we're actually doing okay. I mean, we've just gone over our intended 10 past 11 mark, but I think we have time for maybe Carmen's 2 questions, which she dropped in the chat. And then we can let people take a break, and then we'll reconvene at 11:35 for Alexandra in the next session. So Carmen, there were 2 questions from you.
Yes.
Do you want to go ahead and?
Well, yes. Okay. So actually, John, I missed something about why sometimes you got a burstlet and sometimes you didn't, even though the stimulus looked the same. Maybe there's, and also I also modeled TRPM4, and I think it's not clear how much calcium is required to activate it, but I go with a nanodomain to get up to micromolar.
Okay, so we are not, to start with the second part, right? We are not looking at spatial domains, right? We're treating this as well mixed. So that might be an interesting comparison point between our work. So basically the calcium has to build up enough to trigger enough release and so on, right, to get the significant surge in ICANN and therefore in the postsynaptic voltage. And so the fact that the calcium dynamics, have this slower time course to them is important here. And so there's a lot of dynamics going on under the hood as far as sort of how many cycles it takes for the calcium to build up, or does it ever build up enough to get the recruitment that switches, that actually pushes things into a full burst. And then once the full burst happens, the feedback excitation from the followers to the bursting neurons helps sort of solidify and strengthen the overall output. So was there another part?
The bursting mechanism. What's the bursting mechanism that you wanted to? You said there was a ramp. Is it a burster or parabolic? Or?
So I'm not aware of bursting with a ramp like that at the single neuron level. And so we realized we could get that by having actually taking into account the dynamics of the potassium concentration, right? So in fact, if the potassium concentration is low, you get a sort of slow tonic spiking, and then you get a buildup of external potassium through that outward current, and that gradually transitions things into like this full-blown burst. So that's a sort of separate piece of work that then we incorporated into the network.
Okay, well, thank you. Great talk.
Yeah, thanks a lot, Jonathan, and thank you very much, Carmen, for chairing this series of talks. So just a reminder, everyone, you can take a bio break for now, and we'll be reconvening at 11:35. See you shortly.
Great.